So now we'll switch gears completely, and we'll do the clicker questions, which is probably not having anything to do with hearts whatsoever. So today's first clicker question is, which of the following sequencing techniques, gives you the most sequence per run? Pyrosequencing, Illumina sequencing, Sanger sequencing with gels, Sanger sequencing with capillaries, southern blotting. Thirty seconds. Ten. 5. Stop. What did we think? Most people seem to think it's the Illumina sequencing. That's correct. It's not in the lecture notes, which are printed, but it's what I said, I think, multiple times in lecture last time. Pyrosequencing per run will give you about 400,000 sequences, which is a heck of a lot of sequence, but Illumina sequencing gives you millions of sequences per run. So that gives you much, much more data to work with. Sanger sequencing with gels gives you about 500 bases, let alone millions of sequences. Sanger sequencing with capillaries, again, it depends on how you do those experiments, but that'll give you tens of thousands of sequences, or at least tens of thousands of nucleotides, but certainly nothing near to millions. And then southern blotting, of course, has nothing to do with sequencing. Usually, there are actually some really cool ways you can do sequencing with southern blots, but we're not going to talk about those today. So our next question, cDNA microarray is a lot like DNA sequencing, cloning a gene, multiple northern blots, multiple southern blots, a polylinker. Thirty seconds. Ten. So most people seem to think it's multiple southern blots. I unfortunately don't agree with you. And that has to do with this question. So it's a cDNA microarray. What does cDNA stand for? Complementary DNA. Complementary DNA to what? Messenger RNA. What mechanism do you use to measure messenger RNAs in terms of blots? You use northern blots, not southern blots. So it looks like there's a group that you guys need to sit next to for the next next set of clicker questions. Yeah, the cDNA microarray is basically looking at messenger RNA production. And that messenger RNA production, if it was just a DNA microarray, then the southern blot question would be correct, and a correct answer, because you're looking at hybridization with different DNAs. But if we're talking about cDNAs, that means you're looking at messenger RNAs. So if you're thinking about messenger RNAs, that's going to be your northern blot for looking at this. And we just talked about DNA sequencing, cloning of genes. You actually have to clone most of these genes to make the cDNA microarray in the first place. And the polylinker has to do with what? Polylinkers? What the heck's a polylinker? It's the piece of one of your vectors where you can put in whatever gene you're looking for. So polylinkers usually have multiple restriction endonuclease sites in them. And so that's where you will insert your DNA into a particular vector. So clicker 3, in order to do genetic mapping, you need mating, meiosis, crossing over, markers, or all of the above. .30 seconds. Ten. Okay, this time I agree with you. Yes, if you're doing genetic mapping, you have to have mating, meiosis, crossing over, and markers, all of the above. So that is correct. And so that is one of the big problems that people have with doing human genetics, because humans don't mate with the right other human most of the time in terms of being able to do genetic mapping. So you have to use a whole bunch of other tricks. And I don't know how many of you made it to the biology department seminar yesterday, but it was a really fabulous talk by the chair of molecular and medical genetics from up the hill at OHSU, which reminds me there's also going to be, should be a really good talk this afternoon at 3:15 in the chemistry department seminar series. This is a candidate for a position at the new Collaborative Life Sciences building down in the South Waterfront. Just went over there 2 days ago. It's going to be really, really nice location, and actually people in the back row of the lecture hall actually be able to see me up in front if you don't get overwhelmed by the screen, which is about twice the size of this wall, which is really kind of frightening. But it should be a really nice location down there. So with that, we'll review some genetics. Good luck on your midterms. Everyone has to leave now. Are there any questions, basically, on the stuff that we talked about last time? So for DNA sequencing, there are really sort of 3 different techniques that we talked about. The Sanger sequencing, which is dideoxy chain termination sequencing. Pyrosequencing, which is detecting the pyrophosphate, which is being generated basically by a light reaction. And then Illumina sequencing, which is incorporating fluorescent nucleotides as you go along. And the key with that, particularly that last one, the Illumina, is the optics, because you're looking at lots of little spots on your array, and literally it's like a microscope slide. And it's literally those millions of spots where you're getting all of the sequence data from. Then shotgun sequencing is just the concept whereby what I didn't ask on that first clicker question today was how long any of those sequences are that are generated, because it turns out it varies a lot between the different techniques. The Sanger sequencing actually gives you the longest individual sequence read. It's usually about between 500 and 1,000. The pyrosequencing reads are a couple of hundred if you're lucky, and the Illumina reads are 100. But if you've got millions of them, that makes up for it. And what you can do in the computer is then use the computer to stack all of these sequences on top of each other, find the bits that overlap. And so you have really small pieces of your genome, you get lots and lots of sequence. As long as you've got enough sequence, you can line them all up with each other, and that helps deal with the sequencing error issues that we talked about last time. Blots, okay, hopefully southern blots, DNA, northern blots, RNA. What are western blots? Western blots are proteins, exactly. What about southwestern blots? DNA binding proteins. We'll talk about those later on. So, and people do use them. I've actually, me personally, done that. Talk a little bit about reporter genes, and the idea here is that this is a way that you can look at gene expression. And again, we'll talk much more about this after the midterm. Microarrays we covered. Expression of individual genes. Basically, this is putting your favorite promoter in front of your favorite gene, getting into some organism, usually for some unsuspecting E. coli, to make a lot of whatever protein you're looking for. Then we talked a little bit about human genetics and mapping and knockouts. We did not talk about knockdowns, and that's why I included it in this review. So we didn't talk about it. It's not going to be on the midterm. So today, I apologize for those of you who downloaded the PowerPoint last night. I just realized this morning I didn't put an outline in it. So I will be uploading this with the questions. I do this all the time, hopefully the day of the lecture, to get the updated version that I actually ended up using for class. So today we'll talk about E. coli chromatin. This is unfortunately not in the textbook, but it's exactly the same problem that bacteria have that we have with our DNA. DNA in and of itself is really inefficient. You need 3 bases to encode any amino acid. We're going to talk a lot more about introns and exons. So we have this huge amount of DNA, again about 2 meters long, and it has to get packed in each of our cells. Same problem, E. coli, even though it has a lot less introns and exons, it still has to squeeze its DNA into basically a micron length and half a micron diameter cell. So we'll talk a little bit about that. Talk about how inefficient eukaryotic genes are, again, mostly introns and exons. And then some of the things that have happened in our genome and other genomes, mostly having to do with repeated sequences. And this is one set of repeated sequences. We'll talk about the other set of repeated sequences on Monday, which are the transposons. And then a lot of these repeats lead to gene families, et cetera, which we talked about a little bit before, but we'll cover again today. So as I mentioned before, DNA is pretty inefficient. Again, it's 10 base pairs per turn. This long linear molecule. You could encode a lot more information if you had branched, you had some more compact. Proteins actually can encode a lot more information in a lot shorter space, but probably because of the advantage of having that backup copy with the opposite strand, and probably just what's happened from evolution is what we have now. So if you think about the E. coli genome, it's about 5 million base pairs in length. It's about 1.5 microns in length if you stretch it all the way out. And that's a little over a centimeter, centimeter and a half. But of course, the E. coli itself is only a micron long and half a micron in diameter. So got to compact it 1,000-fold. Most people don't think about bacterial DNA being compacted, but again, has to be in a very similar way to human DNA, eukaryotic DNA in general. You have what's called a nucleoid, which is really just that compacted DNA that you can see inside a particular E. coli cell. And so this is that E. coli cell. The nucleoid is all of this white material, which is all the way through here. How does it get compacted? There are basically sort of 2 different ways, or 3 different ways really that this happens. One are polyamines, and it turns out that polyamines are also used a lot in compaction of eukaryotic DNA as well. Basically, all that these guys are is a whole bunch of positive charges Why do you need a bunch of positive charges? And what's DNA have? Negative charge. Do negative charges like to come together? You're trying to compact things. Negative charges would be a real problem. So you need a counter ion or counter polyions. And so the polyamines, particularly spermine and spermidine, are those which are used particularly in bacteria, but also in eukaryotic cells for this. Lots of small proteins, and we'll look at some of those small proteins a little bit later on in terms of what they're doing and how they're compacting the bacterial DNA. And it turns out a lot of these things are doing it in a very similar way to eukaryotic DNA. Bacterial DNA is a lot smaller. It's easier to work with. The genetics are better, et cetera. And then finally, and we've talked about this before, very heavily negatively supercoiled DNA. And so just that wrapping, I forgot to bring all my phone cords today, but it's that overwinding is positive supercoiling, underwinding is negative supercoiling. But even though you're going the opposite direction as the way the two strands are wrapping around each other, you still get compaction mostly because of the rise. And so you can see that, in fact, if you just look at bacterial DNA, that's shown here. Here is a piece of bacterial DNA, heavily supercoiled, and you can see all of those extra rides where the double strands are crossing each other. The black line that you can see here is really a case of where you have your bacterial DNA. That's that double strand. So the black line is a double strand. But you can see that in some regions, you will also have no rise in this particular region. And that's probably because of exactly the same problem that you have with eukaryotic DNA as well. You have to compact the DNA in order to fit it into the space that you have, but it still has to be accessible to your replication machinery, your DNA repair machinery, your transcription machinery, et cetera. So basically, wherever you have active DNA, something's happening to that DNA, it's not going to be really massively compacted. It has to be released in some way. And so this is a nice example for bacteria, but you can see exactly the same thing in eukaryotic cells as well. The other thing here to point out is that you can have both relaxed DNA and this heavily supercoiled DNA on the same chromosome and at the same time. And these regions then are separate from each other, because when we looked at those circular chromosomes before, if you unwind, it's going to generate supercoils in that particular piece of DNA, but only if those ends are connected to each other. So you can have separate domains, separate pieces of DNA. Most of that is because they're attached to something, and that's what you can see down here at the bottom. So in cartoon form, this is basically exactly the same thing. Here we've got all those proteins that each of these, usually people call them domains, that they're attached to. And then if you, in the case of this example, make a nick, whereby you now allow all of that twist to deal with the writhe, then you end up with a relaxed piece of DNA. That's one piece right here. In a normal cell, that will be the topoisomerases, which are doing this. And so just a different way of looking at any of these individual ones. So how do you make these supercoils? What you seem to do very, very similar things in bacteria as you have in us, eukaryotic cells. These are proteins of the SMCs family, structural maintenance of chromosomes. So we've got SMCs. They're called SMCs in eukaryotic cells. They're called MukB. I don't want to get into why the mutant is called MukB. But basically, if you have a mutant in MukB, cells don't have a nucleoid anymore. And so they don't have that compacted piece of DNA present inside the cell. And they can't grow above about 30 degrees Celsius. These are really sick cells. Doing the same kind of processes we talked about last time and in the lecture on Monday, that gene has been cloned. We have the sequence of that gene. It makes a relatively large protein, 170,000 Daltons. And if you look at that sequence, that's how we know that it's similar to these eukaryotic SMC proteins. We also have figured out what the structure of this particular protein is, true for SMC proteins and for MukB. It's a 5-domain protein. has an N-terminal region. These are two of them here. A long helical region, a linker region, a second long helical region, and a C-terminal region. So you can almost already see this is sort of can act like a clip. So it clips in an open formation and then can squeeze things together at the bottom. And so that's exactly how these guys work. The N-terminal region is an ATP binding motif. It has to come together with the C-terminal region in order to hydrolyze ATP. And so you have to have this closure event to actually have ATP hydrolysis take place. Each of these, so the dark green is one subunit. This brownish one is a separate polypeptide. Usually these guys are folded together where you have just like that long region that I showed between the two, that domain, actually forms a coiled coil, which is also known as the leucine zippers. So every 7 amino acids, you've got a hydrophobic residue which sticks out and interacts with the other subunit. Yeah? Yeah, so there's the N-terminal domain, there's the C-terminal domain, there's those two long helices, and then there's the linker region. And so each of those counts as a domain. Yeah, so each of these proteins, again, with those 5 domains, here these 2 domains are stuck together. These 2 domains are wrapped around each other as a coiled coil. And then we have this linker region. Turns out these linker regions interact with each other. So you can have dimerization. So this will be quaternary structure that forms between these 2 guys. And this is the structure which is usually associated with DNA. And how do we know this and what's actually going on? This has been some developments experimentally, literally in the last couple of years, where people look at literally single molecules of proteins or DNA, et cetera. Lots of really neat ways to do that, but if you're interested, we'll talk about them in office hours. But basically, if you have a single piece of DNA and you add one of these SMC proteins to it, in this case it's MukB, then it will bind to this particular piece of DNA and compact it. And the more of these SMC-like proteins that you allow to bind to this particular piece of DNA, it'll get compacted more and more and more. How does that actually work? It appears to be through really a compaction process where I talked about those ATP hydrolysis when these 2 pieces come together. But really, the important thing is how you get more of those SMC-like proteins added to a complex. Just one is not going to do you any good. You've got to compact the DNA in E. coli 1,000 times. Eukaryotic cell, it's tens of thousands of times you have to compact the DNA. So here we have the MukB protein interacts with other proteins that give you exactly the same phenotype when you mutate them. No nucleoid. Cells can't grow above 30 degrees Celsius. These seem to be bridges between the MukB monomers, and basically they pull the 2 SMCs together and allow another one to form between them. And so if you see here, we start out with these 2 MukBs. They interact with MukE and MukF. That pulls them together in such a way you can get another dimer of MukB, et cetera. So that's all sort of compacting your DNA. And as we'll see in terms of eukaryotic chromosomes, a very similar kind of thing is happening there. So we talked about these so-called DNA bridging proteins, basically binding to different pieces of DNA and pulling them together. Also are lots of DNA bending proteins. These should sound really familiar from the eukaryotic system. Hopefully everyone knows about histones and nucleosomes. Well, these are sort of the bacterial equivalent of nucleosomes and histones. There's this so-called HU protein. HU stands for heat unstable, and again, it's like the classic mutants. You name something after the mutant phenotype. So just like you have with the MukB, things are not stable at high temperatures. You knock out one of these DNA binding proteins, it's no longer stable at high temperature. There are a couple of other proteins, so-called FIS and IHF. We'll talk about these when we talk about how you get transposons and mobile DNA moving around. Turns out that FIS is critical for one of those kinds of transposons, and IHF, integration host factor, turns out to be critical for lambda bacteriophage. That's what this host factor is all about here. And so we really quickly skimmed over those lysogenic, what happens in a lysogenic virus. It integrates its genome into the host genome. And it turns out you have to have this particular protein, the IHF protein, to allow that integration to take place. So this is just an image of that IHF protein bound to DNA, and you can see that it causes this really big bend that takes place in the DNA. Now, the quick thing is that IHF is actually binding the minor groove rather than the major groove. What's one of the big differences between minor and major? Specificity, exactly. And so what does that mean about IHF? Does it bind specifically or non-specifically? Relatively non-specifically. And so that's another thing that you're going to want to have in some kind of chromatin protein. You want to be binding non-specifically to DNA so you can compact all of the DNA, not just a very specific piece of it. So that's it for bacterial chromatin. Now switch gears and talk a little bit about human chromosomes. This is one of my favorite images of the human chromosomes, basically showing binding of fluorescent nucleotides. And so this is a hybridization assay. All these different sequences are going to have different colors, and you can identify all of the chromosomes relative to each other. It gives you a good idea of the variability in the size of human chromosomes. Go from chromosome 21, which is really relatively small here, about the same size as chromosome 22, up to chromosome 1, which is about 250 million base pairs in length. So big difference in terms of the sizes here. This particular, what they call karyotype from, male or female? Female, because we've got two X's here. Reminder that we've got 22 of the so-called autosomes. And so these are the non-sex chromosomes, and then the sex chromosomes here as well. If we look at these, basically in a nucleus, not when they're undergoing mitosis. These are the mitotic chromosomes, where they're the most compacted. The less compaction taking place there, the so-called interphase chromosomes, when you actually can't see them under the microscope. These are highly folded as well. They still have to be extremely compacted. These are loops, which basically most of your DNA, most of the time, are these so-called 30 nanometer fibers where nucleosomes are stacked on top of each other. And this is a reminder to go back and look at that animation that I also posted the link to on YouTube. We look at replication, where you've got all these proteins bouncing back and forth. The beginning of that particular video, and I'll put the link up again, also shows chromosome compaction. So you can see a lot of these, particularly the 30 nanometer fiber formation. So most of the cell, most of the time in the cell cycle, your chromatin is going to be in this 30 nanometer filament form. However, if you need to do transcription, replication, etc., then this 30 nanometer fiber is opened up, giving you so-called beads on a string, because that's what they look like under the electron microscope. These have all the nucleosomes, and then, of course, around the nucleosomes are wrapped our individual DNA. So that's what the DNA itself looks like. What's in that DNA, particularly in the case of the human genome, we've got some single copy genes, a relatively small number of single copy genes, lots of genes that are present as gene families, and so these are related sequences relative to each other, and we'll look at those towards the end of today's lecture. Tandemly repeated genes, these are mostly a lot of the sequences that are right next to each other and multiple times. Why do you want to have multiple copies of individual genes? Backup is a good answer, but turns out you need some proteins, and particularly RNAs, you need very large amounts of them. So if you think about ribosomal RNA, you need a lot of ribosomes. So you have to have a particular piece of DNA which is going to encode that RNA. But one copy is not enough. You end up needing multiple copies. And the same thing is true of the histone genes, which are important for forming those nucleosomes. You need multiple copies of those genes just in order to be able to get enough protein. So those are what most of these tandemly repeated genes are. Introns, again, these are the pieces of DNA that are between your coding sequences for proteins. Lots and lots and lots of repeated elements that we have. Either short repeats that we'll talk about, mostly forming from slippage during DNA replication. Transposable elements, again, that we'll talk about next week. And then extra DNA, which we really have no idea what it's doing. So first, let's look at introns and exons. Again, up here at the top, exons are protein coding sequence. Introns between them, this is really completely off scale. This scale down here is much better. The double black lines are representing your introns, or pieces of DNA that are actually not coding for protein. You can pop these exons together, and you end up with your messenger RNA that's going to encode your protein. And this is one of the reasons that doing those cDNAs are so critical. The cDNA basically is just looking at exon sequence. It's not looking at all that extra intron sequence which is present there. So why bother with all this extra DNA, the junk DNA? Well, A, it's not all junk, which we've just been learning recently. But B, probably it allows much more raw material for evolution to take place. And the way that that seems to happen is either in duplication of genes, which we have down here at the bottom, or even duplications of exons. And it turns out that exons are usually about the same length in many cases as a domain. And so domains are going to fold up. They're going to have a particular structure, which of course gives a particular function. So with those individual functions, if you've got two copies of something that has a given function, you can mutate one of them and maybe get some new function. And so it gives you a lot more raw material to deal with. How does that seem to be happening? It usually seems to be through homologous recombination that happens not with perfectly paired chromosomes, but chromosomes that are offset a little bit. And since we have so many repeated sequences in genomes, when you have a double-stranded break, which can be happening either in meiosis or through DNA damage, it's looking for a sequence which is homologous or very similar to it. We got lots of repeated sequences that could be lining up exactly perfectly, or it could be a little bit off. And particularly think about compaction of DNA inside a nucleus, or for that matter, even inside a cell. You get a whole bunch of DNA all over the place. So this recombination, it's actually surprising you don't get a lot more of this kind of recombination going on. So you have recombination that takes place in this particular case. We have that homologous recombination. You end up with multiple copies of exon 3 in one of your chromosomes, no exon 3 in the other chromosome. If it turns out that you need exon 3, only the organisms which pick up that one chromosome are going to survive. The other ones will not. But now you've got an extra copy of that exon that can be mutated and potentially give you a new function. Same thing is true for genes, and that's just down here at the bottom. Again, lots of repeated sequences. We'll talk about the L1 sequence next week. But repeated sequence, again, you can get this also known as unequal crossing over. We end up with two copies of the gene versus a chromosome that's completely lacking it. This particular cell is fine because it's got two copies of the gene. But as soon as it undergoes meiosis and you lose one of those copies, that one that's missing that gene is probably no longer going to be viable. So we've got these particular gene rearrangements that can take place. They do happen. In fact, surprising to me that they don't happen a lot more often. It turns out there are control mechanisms to make sure they don't. But when you've done these kinds of gene duplications, it allows you to basically regulate the genes, because if you think about our kind of reproduction, what happens? You go through meiosis, you've got gametes. Those gametes come together in an individual cell, fertilized egg. That one egg with one genome, or one pair of genomes in it, develops into the whole organism, which is great, except that different parts of you do different things. Now, your stomach is doing different things than your eyes. And so, but they all have the same genetic material in them. And so how do you deal with that kind of different genetic material? It turns out it's a lot easier to do this if you have introns and exons, because you can mix and match. You can pick which exon you're going to use here, what exon you're going to use there, etc., given those individual different cell types. And if you're interested in more of this, take the developmental biology course. Dr. Litterschmidt talks a lot more about all of these details. Just want to talk about a couple of ways that you can do this. Again, this is our gene up here at the top. We've got two different so-called control regions. You can think of these like promoter sequences. Again, we'll talk more about this after the midterm. And then in this case, 3 exons. So in your simple transcription unit, which means you're using all of the exons, you just are using one of these control regions. This will be a messenger RNA that's going to be the same pretty much everywhere that it's expressed. And the globin genes are a really nice example of that. You don't have much alternative splicing. Probably 60 to 80% of our genes actually have alternative splicing. And so that may explain why we're more complex than something like yeast. So here you can have some cases where you're going to include one exon, and some cases where you're not going to be including that particular exon. You can also have cases where if you include one exon, you don't include another exon. And the way that this works is that you have a polyadenylation site at the end of each of those exons. So if you use this first exon, you're going to be putting on a poly A tail right here. Once you put on the poly A tail, you're not going to be going to this following exon. Or you can flip things around. You can have a different start site. And here, and this is from textbook, it says different cap sites. But you can also just have a different promoter site. So say in one cell type, you're using this promoter. In a different cell type, you're using this promoter. So you end up with a different set of exons, so a different 5' end of your messenger RNA. And what's that going to do in terms of your proteins? It's going to be a different what kind of end? Say different. Okay, we got an N and a C terminus. 50% chance. So that's the N terminus. NC, well, they go both. Hey, someone's going to be right. So no, it's the N terminus. So because you're always going from 5' to 3', and when you translate, you're going to sit down at the 5' end and translate across. And translation is always going from N to C terminus. So in this case, where you have different poly A tails, you have a different C terminus. Here, different starting exon, you're going to have a different N terminus. But still, these guys all share, even this simple transcription unit up at the top here, exons. And so they all have some sequence which is going to be identical. And these are often called isoforms of one particular gene sequence. So that, again, gives you a bit of an idea, and it turns out that alternative splicing can give you up to 10,000, or actually in one case about 30,000 different variants from one given gene. So 30,000 isoforms of one given gene, which is pretty amazing. So we have about 25,000 protein coding genes in the human genome. If you can do 30,000 alternative splicings on each of those, you can see the amount of diversity that you can particularly get in that number of genes is extremely high. Now, admittedly, the 30,000 alternative splicing is an extreme at one end. So got about 25,000 protein coding genes in the human genome. At first, I was really surprised at this table because it says 55% of the human genome, and I always say it's only 1.5% of the human genome, which is really doing anything for us. Well, that's because it's only 1.5%, which is protein coding, so it's exons. So the actual exons are extremely small relative to everything else. But it turns out a lot of these exons are in pieces of DNA that are relatively big. And so introns can be huge. tens of thousands of nucleotides in length, whereas exons are usually on the order of 150 bases. And 150 bases gives you how many amino acids? 50 amino acids. How big are most domains? Most exons are domains, about 50 amino acids. So about 50 amino acids seems to be where you've got exons and also where you've got most domain sizes. So again, protein coding genes, most of which are going to be your intron sequences. Then we have some tandemly repeated genes, and again, these are particularly things for things like our ribosomal RNAs that we just need a whole bunch of copies of, because with transcription, you can get RNA, and that's an amplification of the DNA copy you have. But normally, when you think about proteins, you also get amplification by translation. Well, if you're doing just transcription for functional RNAs, you only get one level of amplification. So you need many, many copies of these things. Then there are a whole bunch of repetitious DNAs. This is present now from the human genome. Simple sequence DNA repeats. We'll talk about these in just a second. DNA transposons, particularly these retrotransposons, where there are literally millions of copies of these retrotransposons that are present inside the genome. Of those, the lines and signs, the long interspersed nuclear elements and small interspersed nuclear elements, which are, again, transposons, comprise the largest part of that. And between the two of them, they're about 34, 35% of the genome, and in some cases, people say up to about 40% of the genome. So if you add all of this together right here, 8, 21, and 13, we'll see when we talk about transposons next week, they replicate a lot like viruses replicate, particularly retroviruses. So I always like to say, because I'm virocentric, that, hey, 1.5% of our genome is encoding protein sequences, but 40% is viral. So we're basically way more virus than we are person, as far as that's concerned. So again, let you think about that. So that's curious, because it turns out that humans actually have way more of these virus-like repeats in them than something like yeast. So we're actually more viral than yeast, which I think is also a bizarre concept. Just take a look at that. We now know the sequence of the human genome. We've known the sequence of the Saccharomyces cerevisiae, standard Baker's yeast genome, for much longer. If you just look at protein coding sequences, this is a classic example here. Exons, again, really small relative to the introns. And these are actually the introns here, and the globin genes are still a little on the small side. They should be even smaller, relatively speaking. So here, lots of non-coding DNA. In Saccharomyces cerevisiae, very little non-coding DNA. And also very few introns. It's mostly intergenic space in Saccharomyces cerevisiae. The other thing to point out here, and this is particularly true in humans, but also a lot of the organisms with larger genomes, is that there are many of these so-called pseudogenes. And a pseudogene just seems to be the result of that unequal crossing over event that took place and then mutation that happened. And it turns out that particular mutation made an inactive gene, and we can tell that it's an inactive gene. There's a stop codon maybe right in the middle of it. There are clearly problems with those genes. This is actually exactly what you would expect if evolution is taking place through this process of domain duplication, gene duplication, and then modification, through mutation and natural selection. You'd expect to have some pseudogenes. If you don't have some selection to get rid of them, apparently our genome can get pretty big. So you have a bunch of these pseudogenes around. Presumably in something like yeast, these are selected for really rapid replication, and replication not just in the DNA polymerization sense, but making more cells. And so in this case, it's probably disadvantageous to have lots of extra DNA around, whereas for humans, it's not a big deal to hang on to all these extra DNA. So that's looking at gene density. We now switch over and look at some of these non-coding RNAs. We already talked about the ribosomal RNAs as having many, many copies in the human genome. tRNAs, again, not surprisingly, again, functional RNAs, lots of copies present in the human genome. There are probably way more of these miRNAs here. So these are microRNAs. These are important for gene regulation. And we'll talk about them, again, sort of right towards the end of the course. And this is a good place maybe to bring up we is actually not going to be me, because unfortunately I'm going to be out of town for about a week and a half towards the end of the term. So I have a couple of guest lecturers. They're listed in the syllabus. Dr. Bartlett and Dr. Raghavan are going to be talking about some of these small RNAs towards the end of the term. But these numbers, particularly these microRNAs and a number of other RNAs, have become just really amazingly prevalent. Again, we talked about the ENCODE project. If you look at how much of the human genome is actually being transcribed and made into RNAs, it's about 80%. So there's a huge amount of RNA, huge amount of small RNAs. So this table, even though it's from a textbook published last year or the year before that, is completely out of date for a lot of these smaller RNAs. So let's look at some DNA repeats. Again, transposons we'll talk about next time. Short sequence repeats. You have a lot of these in the genome. Turns out that they're extremely useful for genetic mapping studies, and particularly these so-called microsatellite sequences. They're usually from about 1 to 13 bases in length, and you have multiple, but most importantly, variable numbers of these sequences here. You can get more copies, you can get less copies every time you replicate your genome. Sometimes these guys are in transcription units, i.e. leading to genes, and we'll see there are a number of particularly neurodegenerative diseases that happen because of this. They're, however, still less prevalent than a lot of these transposons. How do these seem to happen? Basically, in replication, if you already have a whole stretch of otherwise identical sequences, so here this is a trinucleotide repeat, CAG, 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 CAG. When DNA polymerization is happening, it appears that in some cases this guy will just slip backwards, and you end up base pairing here. One of these, or in fact multiples of these repeats, can be lost, and then you can continue to sequence. Now if you copy this strand, you're going to have an extra repeat sequence, and so on and so forth. And so this does happen. How do we know that it happens? Well, it turns out it happens in a number of different human diseases. This particular slide here shows Fragile X, which is a repeat expansion disease. Talk about Huntington's in just a second, myotonic dystrophy, Fragile X, etc. So there are a number of these nucleotide expansion diseases. Most of these are encoding sequences, which then lead to lots of problems. Now, Huntington's in particular is probably the best studied of these. Turns out that there's a trinucleotide repeat. So what do 3 nucleotides do? They code for 1 amino acid. So what happens when you've got lots of extra repeats of this trinucleotide? Changes the sequence of the protein, which eventually is going to change the structure of protein. In this case, it's a glutamine, which is the repeated sequence. And so you end up, or the glutamine codon, I should say. So you end up with polyglutamine in your Huntington protein as it's made. And it turns out with every replication, you end up usually with more and more of these copies. Turns out that that particular protein now precipitates because it can now bind to itself, and it's that process which seems to cause neurodegenerative disease. Now, how exactly it happens, nobody's really sure. May actually be similar to some of the iron deposition diseases that we heard about yesterday in the biology seminar. It turns out that it deposits in some of exactly the same places. So these are repeat expansions which are definitely not good. On the other hand, and this probably depends on your point of view, lots of these repeats are useful if you're trying to do paternity determinations or crime scene investigations, forensics. And that's because these repeat sequences seem to change really quite rapidly. And in fact, between individuals, you're going to have lots of these different repeat sequences in different numbers. Well, so how do you detect this? Well, the basic way to do it is with PCR. So you do PCR, you have sequences which are outside these repeated sequences, and you just amplify across those repeated sequences. So you'll end up with lots of different pieces of DNA, which you PCR amplified, that are going to be different lengths because of the insertions and deletions of these individual nucleotides which are there. And then all you need to do is separate all of these pieces on some kind of gel, could also be capillaries as you use for sequencing, and then just look to see, in this particular case, so mother and child, and then two potential fathers. You just literally go through here and find which of these bands match to which of the fathers. In this case, we've got one here, we've got one here, we've got one here, we've got one here. Number of these sequences are clearly also coming from the mother, but looks like dad number 2 is out of luck in terms of saying that this child is theirs. Same thing is true for forensics. Lots of times these short sequence repeats are used to identify samples which have been found in a particular location. Again, it's a schematic here. The important thing with this is if you just take one of these, statistically it's not going to be that convincing. But as soon as you have multiples of these, then it starts to be a much less likely to be a chance occurrence that you have one of these things going on. So the more details you can have here, the better. So any questions about sort of short sequence repeats, et cetera? Because I wanted to talk a little bit about genome evolution, which is a totally different subject here for the last 15 minutes or so. Okay, so we already talked a little bit about phylogeny. Basically, all that phylogeny is looking at how different organisms or different sequences are related to each other. With this relationship to each other, you can actually put together what you think is, and this is an estimation of what the history of that particular organism or that particular sequence happens to be. If we're talking about molecular phylogenies, this depends on a molecular clock to actually put a date on when you actually have had a family tree or a evolutionary tree has actually happened. And what happens if you look at some of these sequences, purifying selection, genome reduction, and then some of this variation as well. We're probably not going to get to the human genome here until the end. So if we look at a classic phylogeny here, this is now just a primate phylogeny. We look at humans versus chimpanzees versus gorillas versus orangutans. We have a fossil record that goes back about 15 million years. And this is nice because these are organisms that we do have decent fossil records for. And so through the fossil record, we think that humans and chimpanzees diverged about 5 million years ago. But we can also go and look at just nucleotide substitution rates. And about 0.5% nucleotide substitution is about the difference between humans and chimpanzees, slightly more humans and gorillas and orangutans, et cetera. So it turns out that sequence differences actually are really pretty close to the fossil record in terms of how long it seems that these changes have been allowed to take place. So a molecular clock, which is saying, okay, percent nucleotide substitution over a certain number of years, is actually pretty reasonable for relatively closely related organisms. As they start to get further apart, becomes a bit more of a problem. So the idea here is that the sequence difference is proportional to the length of time that these two organisms have actually diverged from each other. And if you think about it, we just talked about mutation rates, so it makes a lot of sense that this could be the case. So let's look at an example of that. This is now the leptin gene. It's important for fat metabolism. If you look at now the human sequence and the chimpanzee sequence, there are a couple of nucleotide changes here. It's the human sequence, it's a CAA. The chimpanzee sequence is CAG. Here, human sequence is AAA, chimpanzee sequence is AAG. And so that's that, approximately 0.5 to 1% difference that you have at the nucleotide level separating these two particular sequences. Now, what's interesting is that most of these sequence changes are so-called synonymous substitutions, and that is that you have a nucleotide substitution, but you don't change the amino acid sequence. So again, if you look here at the chimp protein versus the human protein, here, both glutamines, even though there's a nucleotide change here. Here, lysines, both the same. The only place where you actually have a change here is valines versus a methionine. So even lower protein sequence changes than you have in nucleotide sequence changes. And basically, what that means is there probably was selection against any of other changes that could have happened. Selection against any kind of amino acid change that took place. And it means that it's been a relatively short period of time since these sequences have diverged from each other, and also since those organisms have diverged from each other. And so this is just one example here between these two different sequences. You can put together a lot more sequences here and basically say again here, gorilla, chimp, and human are all very closely related to each other. We're quite distantly related from opossums and wallabies. These are the marsupials. The non-marsupial mammals here. All of these, curiously enough, have about 3 million base pair genomes. Why that is not entirely clear, but they all have about the same size genomes. And it turns out that it's a lot easier to get sequence from all of these currently extant organisms than it is to try and look at differences in morphology, differences in skeletons, etc. And sequence data is giving you also really pretty nice trees of how these things are all related to each other. And in fact, there's been a real resurgence in terms of phylogeny and taxonomy. People think about how organisms are related to each other based on sequence now, instead of a lot of the morphology and the way that classically you've classified organisms. So I think this has really been kind of a revolution in terms of thinking about a lot of these sequences. One really nice example of this is if you look at humans versus mice. And again, well, let me back up here. I think the mice were on here somewhere. Yeah, rat and mouse are right here. So they're related, but relatively distantly related to humans. But rats and mice are really good experimental organisms, particularly if you're talking about genetics. You can get the mice to mate with who you want them to mate with, unlike humans. Usually don't do that terribly well. So nice about mice is that it turns out that you can also use these molecular clocks for looking at how different they are relative to each other. What's neat is it looks like there's selection clearly going on in exon sequences. Again, these are things that are coding for proteins, whereas in introns, it's all over the place. Sequence similarity is actually really hard to detect. And basically what that means is in exons, you're having selection because there's some function. Again, that protein is leading to some function. If you change the protein sequence, then whatever organism it is or mutant it is that has that change is not going to survive. So it's be selected against. On the other hand, introns, nobody seems to really care. A lot of this stuff seems to be pretty standard, and so if you just look at intron changes between something like humans and mouse, you can get a pretty good idea of what the rate of mutation is, whereas if you look in exons, that tells you what kind of selection you're actually having. So these are all then relative similarities to each other, and this is a great way of actually looking at these sequences. Here again, it's a mouse sequence at the top, human sequence at the bottom, exons. These things are extremely similar to each other. As soon as you get into an intron sequence, sequences are all over the place. And this, as it turns out, is a really good way of detecting what an intron is and what an exon is. You just get the sequence of the mouse genome, you get the sequence of the human genome, you compare them to each other, and then you find the areas where the nucleotides are more similar to each other. You say, oh, those are likely to be exons versus the introns, which are right next to them. And it works extremely well. And it turns out when we talk about splicing later on, you'll see that trying to figure out what is an intron and what's an exon is sometimes really hard. And so using this evolutionary approach to be able to look at these things turns out to be a really powerful one. It's not just the case for looking at introns and exons that humans and mice are pretty similar. it turns out that we've got almost the same sets of genes, but they're all on different chromosomes relative to each other. So if you just look at the human chromosomes, this is another basically same as that image I showed with the karyotypes, only we're now looking just at haplotypes. Again, these are the autosomes 1 through 22 and the X chromosome. All of these individual genes here, color-coded, are color-coded relative to where they are on the mouse genome. Turns out almost all of the genes that we have on our X chromosome are also present on the human X chromosome. It's probably because there's a lot of selection in terms of these sex chromosomes. On the other hand, you find that particularly for some of our larger chromosomes, these guys have been mixing and matching all over the place. And this is probably, again, what's happened in ancestors. And as we have evolved from those common ancestors, in the case of mice, probably a couple 100 million years ago, there's been lots of genetic exchange between these different chromosomes, but still, a lot of those same genes are still there because there's selection for function, but very little selection for where these things happen to be on any given chromosome or any of these particular places. So if we look at one of those chromosomes, this is mouse chromosome 12 and human chromosome 14, basically, we've got these regions of so-called synteny, and synteny is just same genes, same order. But, big pieces of sequence like this one here, which are either have been lost in the development of mice or gained in humans. Most of those turn out to be transposons that, again, we'll be talking about on Monday. So this is, great, allows you to understand a lot about mice versus humans, although, as we heard yesterday in seminar, mice are not really good models for neurodegenerative diseases because they're hyper anyway. But it turns out that there's one particular organism that is really strange relative to something like humans and mice. And this is, of all things, the Fugu fish, the puffer fish. Now, why it has a much, much smaller genome than any of these other ones is completely unclear. But it does. And so it's only about a 0.4 gigabase pair genome, so 400 million base pairs, as opposed to ours, which is about 3 gigabase pairs, and true for most fish as well. So what's the difference between this puffer fish and us? Well, turns out it just has a lot fewer repeat sequences in it. Again, why is not entirely clear. Lot fewer introns, lot fewer repeat sequences, but exons are basically identical between the two. And so if you're going to think about doing that kind of thing I was talking about with mice and trying to find exons and introns, trying to find exons, sequence the Fugu fish genome. And in fact, the Fugu fish genome has been sequenced. And it's a really nice way of identifying where you've got exons and introns, because you can find this, in this organism, Fugu, you've got very few introns. And so you can now use what you found in Fugu now to go back to studying the human genome and taking a look at that. So I just finish up with this last slide, which has to do with taking advantage of all of these genome sequences from all kinds of different organisms and then literally using them to find exons. And so here we've got Fugu down here at the bottom. Here, nice sequence here, similar to all of these other organisms. And it turns out that's an exon. So great way of identifying exons. If you look at chimpanzees, orangutans, and baboons versus the human genome up here, worthless in terms of finding introns and exons. There are, however, some actually really bizarre things that people have found in looking at some of these multi-organism alignments. And that is, if you look at something like an opossum versus chimpanzee or human, you find some of these sequences, like this one right here, that one right here, and that one right here. They're conserved in a whole bunch of different species, but they're not in exon. So what does that mean? I'll let you think about it over the weekend.